the current work at my lab on DNA-based cancer identification and targeted drug delivery. So first I'll talk about implementing a cascading chain reaction circuit that operates on the surface of various cancer cell lines for precise identification. And in fact, it's I believe it's the most precise identification of cancer cell lines. And then second part of the talk is on a peptoid-coated DNA origami capsules for safe and effective drug delivery. Have you received the email yet?
Yeah, I'm just about to share it.
It's a PDF.
Yeah.
Okay, so beginning the first section on the current work on DNA-based cancer identification, we implemented a cascading chain reaction circuit where the chain consists of DNA hairpins, and they are attached by hybridization to distinct aptamers, each targeting a specific membrane protein specific to a particular cell type. And so basically, these self-assemble onto the membrane protein specific to the cell line. And then after input of an initiator, they create a chain reaction. And you can either have photonic output, fluorescent output, or it can be transduced to bind to and trigger in DNA capsule for drug delivery. So,
John, can you
yeah,
see
yes,
see the presentation?
Good.
I'm on the page you want me to be on.
Well, go back just one slide. Okay. So yeah. Then the next slide. And on this slide, we're describing sort of essentially the architecture for the cell surface circuits, and the advantages are of the architectures. It's very modular, fast, and relatively low leak. So as I said, the DNA hairpin gate is hybridized to the aptamer, and the DNA aptamer is specific to a particular protein, a membrane protein of a cell line. And so then by this chain, you're really identifying a number of distinct membrane proteins specific to that cell line. And this was published in a JACS article in 2019. And the principal first author was Tianqi Song, who has since graduated and is a postdoc at Caltech. And going to the next slide, this is giving you a close-up of the individual gates. They're basically, they're called nodes. There's a hairpin, and then the tail is hybridized to an aptamer. And now the aptamers bind to specific protein. The membrane proteins of the cancer cells, the membrane proteins freely migrate, and eventually they'll consecutive gates will be adjacent, and an initiator will initiate the first node or gate of the reaction, and there's a chain reaction, essentially a linear cascade. So it's essentially HRC on a cell surface. And going to the next slide, we were able to identify quite a number of different cell lines, and there was relatively low false positives, particularly for the aptamers that had tight binding with specific proteins. Going to the next slide, the system is fairly modular because you can attach distinct hairpins to distinct aptamers and swap them out. And going to the next slide, there's ongoing work where we're working to be able to diffuse in the medium signals that are output signals from these circuits on a circuit on a particular cell type to another cell type. So essentially they can communicate. We're in the process of attempting to demonstrate that, and the same diffusion process could be used to activate capsules and other molecular objects. And then going to the next slide on catalytic variations, there's another scheme where you can float on the, attach via aptamers to cell membranes, both fuel strands and input strands and gates. And that scheme we're in the process of testing. It may have some advantages, but it's a bit more complex. So in summary, we were able to demonstrate these hairpin chain reactions on the surface of cells with the current output is fluorescent output. And going to the next slide, the next slide is sort of mentioning the other part that we'll talk about, assuming we have some time. It's a design of D-Day origami capsules that are peptoid-coated for protection and could be used for example, drug delivery. And so they could be loaded with a particular molecule that acts as a drug and then activated, opened, and a UNC collaborator is producing peptoid coatings, and we've actually already synthesized such a thing, and the peptoid can protect the whole capsule from degeneration and in the serum. And then the next slide is sort of a bit of an image. You can see, unfortunately, it's a PDF, so it's just a single image, but on the PowerPoint, it's a dynamic image of a, in this case, a three-dimensional.
You just muted yourself, dude. Dude, you're muted.
John, I think that you are muted for the moment.
Okay, I'm sorry. So on the next slide, it's a smooth, curved nanostructure. And going back to the previous slide, could you go back to the previous slide? It has the properties that it's, you can design it to have multiple helix thicknesses, multiple scaffolds, and curvature with inflection points that we'll talk about. And the software uses heuristics to find crossover positions. And the whole system is sort of shape focused, where you define a shape. And going to the next slide, there would be a 3D CAD input to the system. So it might be, for example, something you produced, and then the software produces essentially a mesh design, and then it does the lambda sequencing and staple routing, and then synthesis. But it also, you can specify thickness. Going to the next slide, here are, we've done a number of demonstrations in collaboration with Hao Yan's institute and lab. So, for example, there's hyperboloids, there's twisted ovals in the middle, and there's a Klein bottle. The Klein bottle, we're still sort of doing the final demonstration. Of course, it's not a real Klein bottle, but it is, it's sort of a bottle with a hole where the sort of twisting arm goes through the hole back in there. And there's a whole series of other objects that we've characterized experimentally as well. Next slide. The issue is, in some cases, you need to reinforce the assembly, the nanostructure, and that can be done by having double and triple layer sections. Next slide. It should be dynamic, but we've experimented and found that in fact, having multiple layers does considerably help in certain sections, but there's a somewhat tricky optimization problem between increasing the crossover density and the increased number of helices, which does gain rigidity, versus sort of the need for localized staple binding locations. So going to the next slide, this is an image of an experiment on sort of a cone where we've managed to reinforce the top section by having a further layer, and we were able to demonstrate that experimentally stabilize the upper section of the cone. And next section is sort of the use of a special zigzag sort of layout of the scaffold strands to increase modularity. So the zigzag idea allows you to split up the scaffold into multiple scaffolds if you're running out of scaffold length. And so in the next slide, there's the issue of how to deal with inflection points where you're going from convex to concave. And we found that there was considerable strain there in both the simulations and in the experimental demonstrations. So we found that we could overcompensate for higher strain, and then we could actually achieve the targeted shape. And there's some complexity to that. I'll skip over it. Next slide is the real the software has fairly sophisticated heuristic algorithms for variable crossover locations, and the actual architecture of these DNA origami, curved DNA origami, are concentric cyclic structures with the zigzag for the staple, and there's considerable issues to do with issues like helical twist, and sometimes crossovers are not available, and the heuristic algorithm goes back and forth and optimizes this until it's as specified in the 3D input by the user. And so, of course, by adjusting crossover locations, you can make the nanostructure locally curved or linear or convex, concave, and so forth. Next slide is the final slide. Basically summarizing this final slide and trying to keep the talk in schedule, there's a particular sort of challenge when you have, for example, double and triple layered nanostructures. So the crossovers will cross over both vertically, but also within layers. And so we've developed sort of heuristic schemes for doing that. And experimentally, we've been able to demonstrate all these assemblies. So going back to the slide where it had the Klein bottle and so forth, it's titled Rendering of Curved Surfaces. So in the article, we've got quite a catalog of different complex surfaces. The most probably complex is the Klein bottle. And then going back to the start slide, the first slide of the whole talk, just to summarize, we've been trying to transition our lab's work in DNA nanoscience to things of impact to nature. And what I reported today was cascading chain reactions on the surface of cells, work to get it between cells, and then this software suite for three-dimensional DNA origami capsules. And finally, I should mention that our lab during COVID developed an RT lamp protocol, which is we started in early February and ended in October, and was tested for many months by an Oregon virology lab, and it says precise 95% as PCR, and it's a $2 micro test tube with no equipment, just you immerse it in a thermos at the right temperature, and it's colorimetric output, and it's stable for at least a month. We're now testing 2 months at 0 degrees centigrade, and we believe it'll be good for the third world for COVID testing, but I have no slides on that. So thank you very much, and I apologize for the technical difficulties.